Hello dear friends, once again it's very hearty and warm welcome. Dear friends, in this lecture we are going to study first of all about this little topic, that is pollen pistil interaction, and it is followed by the mechanism of double fertilization. In 11th we people have studied little bit about the double fertilization, and you know that double fertilization is the exclusive phenomenon seen in angiospermic plants also. So first of all, let us talk about the pollen pistil interaction. Here the pollen stands for pollen grain, pistil stands for gynation or carpel, and interaction means the reciprocal influence of pollen grain and carpel upon each other. It means here we have to study about the interaction taking place between the pollen grain and pistil after the process of pollination, after the process of deposition of pollen grain on the stigma of carpel. Actually, the pollen pistil interaction include series of events from pollination, that is from the deposition of pollen grain on the stigma up to the entry of pollen tube into the ovule. So let us talk about all those different events briefly under this title, pollen pistil interaction. I said just now that the pollen pistil interaction, it includes series of events from the pollination, that is from the deposition of pollen grain on the stigma up to the entry of pollen tube into the ovule. Now after pollination, after deposition of, we can say, pollen grain on the stigma, first of all the first step is recognition of that pollen grain by the stigma. It means the deposition of pollen grain followed by the recognition of pollen grain, and it is followed by either promotion or inhibition of pollen germination. Try to understand. I said that after the process of pollination, after the process of deposition of pollen grains on the stigma, first of all the stigma it recognizes that pollen grain. It means whether the pollen grains which get deposited here are compatible or incompatible, matching or non-matching, whether that pollen grains are of right type or wrong type. Now during self-pollination, we know as the pollen grains of the same flower or neighboring flower but of the same mother plant get deposited on the stigma, and hence there is no chance of incompatibility, there is no question of non-matching between pollen grain and stigma, and hence immediately that pollen grains are allowed for further process of germination, for further development of male gametophyte. But the cross pollination has no guarantee. It means we know during cross pollination the pollen grain may be of other variety of plant or maybe of other species are deposited on the stigma, and that's why first of all the recognition of that pollen grain takes place, and if that pollen grain found is of right type, matching, compatible, then it is allowed for germination, and if that pollen grain found as incompatible, non-matching, then its germination is prevented. And such non-matching, incompatible or wrong type of pollen grains are, we can say, broken down, they wither off, they are inactivated, and they are not allowed for further process of germination. We have already studied that the pollen grains are at two-celled stage at the time of pollination. So in some cases we know maybe at three-celled stage, but in majority of the angiospermic plants the pollen grains are at two-celled stage. Pollen grains are at two-celled stage during the deposition on the stigma, and such two-celled pollen grain has a small generative cell, haploid generative cell, and a large tube cell which may be called as vegetative cell, tube cell or vegetative cell. Now, after the deposition of right type compatible or matching pollen grain on the stigma, this stigma recognizes the pollen grain. This recognition is possible because of certain protein molecules present in the pollen grain as well as we can say in the stigma, and due to the matching or maybe non-matching of that proteins of pollen grain and stigma, the further process of germination is either allowed or prevented. Now in right type of pollen grain, compatible type of pollen grain, once it get recognized, this pollen grain, it imbibes the stigmatic secretion, that is a sticky secretion secreted by stigma which contain certain minerals, nutrients, water, and the intine of pollen grain protrudes out through the germ pore in the form of tube. In the form of tube, you know, it is known as pollen tube. Now this pollen tube, we know, carries the nucleus of tube cell at its distal end, and at the same time the generative cell now divides mitotically to form two small and haploid cells that are nothing but the male gametes. And now this three-celled structure is known as male gametophyte. Pollen tube formed grows and run towards the ovary. The pollen tube, it grows and runs towards the ovary. The pollen tube pierces through the stigma, style, and run towards the ovary. Of course, the movement or growth of pollen tube towards the ovary and ovule is under the influence of some chemicals secreted by the filiform apparatus of synergid cells. You all are very well known. In this diagram, I have shown anatropous ovule with female gametophyte, and here is a group of three cells, that is egg apparatus, middle larger egg cell, female gamete, and two laterally placed smaller synergid cell. We have already studied these synergid cells show a filiform apparatus and it produces some chemicals. The movement of pollen tube towards the ovule is guided by that chemicals produced by the synergid cell. Means we can say that pollen tube grows towards the ovule under the influence of chemicals, hence it is a sort of chemotropic movement. Okay. Now this pollen tube, when grows towards the ovary and ovule, through the pollen tube it secretes some enzymes such as we can say pectinases and hydrolases. These enzymes bring about the breaking down of stylar tissue and facilitates the growth of pollen tube towards the ovule. In most of the monocot plants and few dicot plants the style is hollow, but in we can say very few monocot and most of the dicot plants as style is not hollow, it is solid, it is slender, and that's why for the proper penetration of pollen tube, passing of pollen tube, the enzymes pectinases and hydrolases are produced which break down the stylar tissue, which facilitates the growth of pollen tube towards the ovule. Now this pollen tube carries the tube nucleus and two haploid male gamete towards the ovary. Mostly this pollen tube enters into the ovule through the micropyle, and then here it bursts off, releasing its content, and it is followed by the process of fertilization, say double fertilization in angiosperm. So here simply we have to study pollen pistil interaction, means the series of events taking place from the deposition of pollen grain on the stigma, then it is followed by the recognition of pollen grain, followed by either promotion or inhibition of pollen germination, up to the entry of pollen tube into the ovule. Here we have discussed about the pollen pistil interaction. Now we have to study about the process of fertilization and double fertilization. Fertilization and double fertilization. We know that fertilization is simply the process in which haploid male gamete and haploid female gamete unite together to form diploid zygote. The process of union of single haploid male gamete. The process of union of single haploid male gamete and single haploid female gamete to form diploid zygote is known as fertilization. Fertilization. Now, we people have to study the double fertilization, because I said that double fertilization, it is the characteristic feature seen in angiospermic plants also. Only angiospermic plants, because it is the unique feature of angiospermic plant. The process of double fertilization was discovered by scientist Navaschin in Fritillaria species, Lilium and Fritillaria species of plants. Let us talk about the mechanism of double fertilization. As I explained earlier, after the process of pollination, after the deposition of two-celled pollen grain on the stigma secretes few sticky secretion, that is stigmatic secretion. Pollen grain absorbs that stigmatic secretion and the intine protrudes out in the form of pollen tube. At the same time, the generative cell divides mitotically to form two haploid male gametes. And now here this pollen tube, it carries that tube nucleus and two haploid male gamete towards the ovule, towards the ovary. In angiosperms, as the male gametes are non-motile. In angiosperms, the male gametes are non-motile. Angiosperms मध्ये male gametes हे non-motile आहे. It means they don't have ability of independent movement, and that's why the non-motile male gametes are carried. Non-motile male gametes are carried up to the female gamete by means of pollen tube. It is known as siphonogamy. Siphonogamy. Remember, siphonogamy means it is the act in which non-motile male gametes are carried up to the female gamete by means of pollen tube. Siphonogamy. Let me tell you, in case of algae and other plants also, if the male gametes are motile, flagellated and can swing and reach independently up to the female gamete by its own, then that act is called zoidogamy. Zoidogamy means the male gametes are motile and they can swim, they can show movement, they show motility just like the animals and reaches up to the female gamete. That act is called zoidogamy, just like the zoo, that is animals. In case of gymnospermic plants, here we are talking about the angiosperms, but in gymnospermic plants, the male gametes are ciliated, means they can show motility, but then and then they are carried up to the female gamete by means of pollen tube. So, in gymnosperm, the act is intermediate stage between zoidogamy and siphonogamy. But here in angiosperm, pure siphonogamy is seen, that is, the non-motile male gametes are carried towards the female gamete by means of siphon, that is pollen tube. I already told you the movement and growth of pollen tube towards the ovule is chemotropic because it is influenced by the chemicals produced by the filiform apparatus of synergy cell. Now, when this pollen tube enters into the ovary cavity and reaches near the ovule, mostly pollen tube enters into the ovule through the micropyle. Mostly the pollen tube, it enters into the ovule through the micropyle. You know micropyle? It is a small vertical slit or opening present at the tip of body of ovule. Let me tell you that the length of pollen tube is variable. In few flowers, the length of pollen tube is only few millimeter and maybe up to 45 centimeters. Say in case of maize, it is very much elongated. Anyway, so the pollen tube mostly enters into the ovule through the micropyle. This process of entry of pollen tube into the ovule through micropyle is known as porogamy. Entry of pollen tube into the ovule through micropyle. Here poro stands for pore, that is opening, micropyle, and it is most common method. Say for example in lily, porogamy is seen. Sometimes the pollen tube may enters into the ovule through chalaza, means opposite end, from opposite end. You know the base of body of ovule is called chalaza, and the entry of pollen tube into the ovule through chalaza is known as chalazogamy. It is less common method. It is seen in Cosorina. And very rarely the pollen tube enters into the ovule along the lateral side. That is, we can say by penetrating the integuments. And the act of entry of pollen tube into the ovule along the lateral side by piercing the integuments is known as mesogamy. It is very rare, seen in cucurbita. Now, when the pollen tube enters into the ovule through micropyle, pushes the nucellar tissue and reach near the egg apparatus. Reach near the egg apparatus. Reach near the egg apparatus. As we know, this pollen tube carries a tube nucleus at its tip here and two haploid male gametes. Two haploid male gametes. Okay. The pollen tube, it pierces one of the synergid cells. We know these two laterally placed cells are called synergid cells, and thus the pollen tube pierces one of the synergid cells, absorbs the water from that synergid cell, hence synergid cell burst off, pollen tube swells due to the absorption of water, and pollen tube also burst off, releasing its content near the egg apparatus. अर्थात हे पोलन ट्यूब या दोन सिनर्जिड सेल्स पैकी कुठल्या तरी एका सिनर्जिड सेलला पेनिट्रेट करते, पियर्स करते आणि त्या ठिकाणी सिनर्जिड सेलही बर्स्ट होते आणि पोलन ट्यूबही बर्स्ट होते. अर्थात पोलन ट्यूब मध्ये असलेले जे कंटेंट आहे, ट्यूब न्यूक्लियस अँड टू हॅप्लॉइड मेल गॅमेट्स are free near the egg apparatus. Now, out of these two haploid male gametes, one haploid male gamete, listen carefully, one haploid male gamete fuses with haploid female gamete, that is egg cell. Female gamete, that is egg cell, to form diploid zygote. Let me show here in diagram. Suppose out of these two haploid male gametes, one male gamete unites with female gamete, that is egg cell, haploid egg cell, to form diploid zygote. Let me show you. Here is diploid zygote. This process of formation of diploid zygote by the union of male gamete and female gamete is known as syngamy. Or first fertilization. While the other male gamete, second male gamete, it fuses with diploid secondary nucleus. You know, here with the help of blue chalk I have shown diploid secondary nucleus. So another haploid male gamete fuses with diploid secondary nucleus to form triploid primary endosperm nucleus. It is abbreviated as PEN, primary endosperm nucleus. And the formation of triploid pain by the union of second haploid male gamete and diploid secondary nucleus is known as triple fusion. Or say second fertilization. Let me show here. Suppose this second male gamete fuses with diploid secondary nucleus to form here a triploid pain, primary endosperm nucleus. It is a nucleus having 3 sets of chromosome, because male gamete is haploid and diploid secondary nucleus is having 2 sets. So togetherly here is a formation of triploid pain. This process is called triple fusion or second fertilization, and both these processes that is syngamy and triple fusion, constitute the double fertilization. So, how one can define double fertilization? Double fertilization is the process in which both haploid male gametes take part in the process of fertilization. Single haploid male gamete unites with haploid female gamete to form diploid zygote, while another haploid male gamete unites with diploid secondary nucleus to form triploid primary endosperm nucleus, and thus this process is called as the double fertilization. So, the product of double fertilization is diploid zygote and triploid pair. After double fertilization, so many noticeable changes occur in the ovule as well as in the ovary, and it leads to the formation of seed and fruit respectively. That formation of seed from ovule and formation of fruit from ovary are known as post-fertilization changes that we have to study in our next lecture. But here, briefly let me tell you what is the significance of double fertilization. Very simple. Double fertilization involves syngamy as well as triple fusion. Syngamy leads to the formation of diploid zygote, and this diploid zygote gives rise to the embryo. Triple fusion leads to the formation of triploid pen, and this triploid pen gives rise to the endosperm, that is a nutritive tissue that we shall study later on. Also, due to the syngamy in the embryo, diploid condition is restored in the plant, in the new plant. Then we can say that double fertilization, it stimulates the formation of seed from ovule and formation of fruit from the ovary. So here we have discussed and completed the study of these two topics, pollen pistil interaction and double fertilization. So post fertilization changes we shall study in our next lecture. Thank you and bye.